So this is the review for Immunology Test 1. As a disclaimer, anything that was mentioned in class or in lab may be tested. Approximately 50% of the grade will come from multiple choice questions and approximately 50% will be from short answer questions. Lab material isn't reviewed here in this video, but it is testable material, so be sure to review your notes from the lab. So what is immunity? It's a state of protection against pathogens that are foreign. Can we generate immunity without inducing disease? Yeah, we can use vaccines, and they've been done since the 1700s. Vaccines have been used to protect or induce a protection against pathogens. What is humoral versus cell-mediated immunity? So when we talk about humoral immunity, it's talking about the fluids and how things can be made and released, such as antibodies. So humoral is talking about antibodies often, or other non-cellular components, so things that are in the blood. And cell-mediated is talking about things that involve most often T cells. How are the adaptive immune cells activated? So in broad terms, when an organism or an animal comes in contact with something that's foreign, so a pathogen, it will mount an immune response. And that to get the activation of the adaptive immune cells, you would need to have presentation of antigen to the T cells. It's not quite shown here, but the T cell is showing you that it gets activated by antigen, and that would be with an antigen presenting cell to activate the T cells. And there are T helper cells which get activated to release cytokines, and those cytokines help other cells get activated. So cytokines that are released from T helper cells activate cytotoxic T cells be activated when they're presented antigen from an antigen presenting cell to activate them. And cytokines released from T cells also help activate B cells. And B cells have a B cell receptor, which is membrane-bound immunoglobulin, and they detect antigen and get activated and then start secreting antibodies. And when they're secreting antibodies, they're called a plasma cell. T helper cells help by making cytokines, which can help activate other cells, whereas cytotoxic T cells or T killer cells, when they're activated, they go out and find infected cells and kill them. What's different with innate immune recognition of pathogen versus adaptive? So when we're talking about recognition, we can think about the receptors that are involved. And so in the innate immune response, there are these pattern recognition receptors that are coded for by genes. So they're germline encoded receptors. And they recognize things that are conserved on pathogens. So they recognize pathogen-associated molecular patterns, which are shortened down to the word PAMPs. Whereas in the adaptive immune response, the receptors are randomly generated to give you B cell receptors or T cell receptors. And they bind antigen, and it's very specific, short amino acid sequence that they're recognizing here. Whereas in the pattern recognition receptors, those are generic molecules that are found on many pathogens that are being recognized. So the B cell and T cell receptors, their receptors are very specific in what they recognize. What is clonal selection and expansion? So when a cell, this is showing you B cells, So there was a stem cell that divided, and it can become any number of different cells depending on the environment it's in. And in this example, it's showing you how there's several different B cells that are generated, and they're different. So they have different numbers because they recognize something different. So the antibody on the surface recognizes different antigens. And then clonal selection and expansion means that only the B cells that get activated with antigen are going to clonally expand, so become more of them if they get activated. If they, through the primary lymphoid organs, they may go and circulate through the body, but if they don't actually interact with antigen, they're not going to expand into more of that same exact B cell. And some, it's showing you here in the primary lymphoid organs how some B cells actually don't make it into circulation because it may be because they make a B cell receptor that's actually recognizing self antigens. So those are deleted in the bone marrow. Why does the immune system not usually respond to self antigens? We don't want to respond to self antigens, but certainly the possibility is there because of that recombination of the B cell and T cell receptors, and how we manage to avoid most often recognizing self antigens is that we have tolerance. And so this is a way that those random rearrangements in the B cell and T cell receptors are removed so that we don't recognize self antigens. So it's the fact that we have the ability to induce tolerance. What are the main differences between the innate and adaptive immune response? So if we highlight a few of the main differences, the timing with respect to the activation of the innate and the adaptive is different. So it's very quick to activate the innate immune response, whereas it takes many days to activate the adaptive immune response. The specificity is different between the two. The innate immune response recognizes conserved patterns on pathogens. So it's a fixed number of things that they can recognize. And the adaptive is highly diverse because of those gene rearrangements that occur in the B cell and T cell receptors. And it can even improve during an immune response. So the B cell receptor, for example, can improve how well it binds during the response. You get the same response each time you come in contact with the same pathogen in the innate immunity. However, adaptive immunity will, if it's the same pathogen, it will respond more rapidly and therefore be more effective each time you come in contact with a pathogen. And that's what we take advantage of when we give someone a vaccine, because if we give them pieces or parts of a pathogen, we want to induce an immune response so that they would respond much quicker if they ever do come in contact with that actual live pathogen. The main components are different between the innate and adaptive. In the innate, we consider barriers such as the skin and chemical barriers such as the pH or acidity, phagocytic cells, cells that eat and destroy microbes, and those pattern recognition receptors that can detect conserved structures that exist on many different microbes. In the adaptive immune response, we have T cells and B cells are the primary cells, and they have antigen-specific receptors. The B cell receptor is a membrane-bound antibody, and a T cell receptor is on the surface of the cell, and it is going to only recognize antigen in the context of MHC. Presented antigen in from an antigen-presenting cell. Antibodies are made by activated B cells. So the plasma cell is a cell that's generated from an activated B cell, and antibodies are released or membrane bound depending on the B cell. How is the adaptive immune response different the second time you come in contact with the same pathogen? So a few things to highlight that are different. You do have a memory response. So the adaptive immune response has memory because there are memory cells that are generated when a B cell or a T cell is activated. And they will circulate through the vasculature and the lymph nodes and the lymphatics to be ready for if they come in contact with that same pathogen a second time. So the first time, the amount of immune response takes several days to be initiated, and the second, so this is the adaptive here in the dark blue, and the light blue is the innate. The innate is the same every time you come in contact with the same pathogen. So it's showing you here that the innate takes some time to activate, and it'll be activated for a while. And it's the same the second time you come in contact with that same antigen. The adaptive takes several days after the first response. And if you come in contact with that same antigen the second time, you're going to have a quicker response, and it's going to be much higher response. So, for example, the level of antibodies that are generated, the amount, is much higher, and often the specificity of the antibodies is much better. And this is because of this memory that you have. What are some problems that can occur with respect to the immune system? So we can have overly active immune systems, and some people have allergies or asthma, as well as autoimmune disease, such as multiple sclerosis or Crohn's. So this is when you have an immune response against something that you don't want to have it against. So you have an immune response against peanuts, or you have an immune response against the myelin sheath in your covering your neurons. So these are immune responses that are not where you would want them to be. You can also have immunodeficiencies, which can be genetic, so that would be a primary immunodeficiency, or you can have an acquired one, which is a loss of immune function that you get due to something that's occurred to you. Sometimes that the immune system are also relevant is when we have transplants. Transplanted tissue, we would want to not mount an immune response to it. So we were trying to avoid immune responses. And the body's natural response is to fight a transplanted tissue unless it's exactly the same. So some people would have the best transplant would be from maybe one location, like one part of your leg, a graft from one area to another on the same individual. Or the next best thing would be from an identical twin. And then if that's not a possibility, you would go to someone else who's closely related, or at least has some of the genetics looking similar. And so that's something that would need to be examined. Another situation where we would want to activate the immune response is in a situation where someone has cancer. So there's cells that are host cells that are now dangerous, and so now we would want to have the immune response target and kill those cells, but they're self cells, so it's hard to do this. We're generally tolerant of self cells, and it's hard to generate an immune response to them. So these are a few examples of where the immune response can be a problem, where it's overactive or underactive given the situation. What can an imbalanced microbiome at times indicate? So it can indicate a problem can lead to inflammation, and you can get different diseases if you have, they see correlations between having a dysregulated microbiome and type 2 diabetes, some mood disorders, and chronic inflammation such as inflammatory bowel disease. So they're seeing that the type of microbes that you are colonized with have impacts on many different aspects of your overall health. Where do the white blood cells come from? So they come from the bone marrow, and they originate as hematopoietic stem cells. And the process of generating the red and the white blood cells is called hematopoiesis. What can the myeloid progenitor cells become versus the lymphoid progenitor cells? So if we look at this kind of tree of where the immune system cells come from, that hematopoietic stem cell can become a myeloid progenitor or a lymphoid progenitor. And the myeloid progenitor can become dendritic cells, or they can become monocytes, neutrophils, eosinophils, basophils, and mast cells, platelets, or red blood cells. And so these ones here in the middle are granulocytes because they have granules in them. So there's a lot of different things that can be generated from a myeloid progenitor. And then from a lymphoid progenitor, you can also get dendritic cells, as well as your adaptive immune cells, the T cells and the B cells. And you can also get an innate lymphoid cell, such as the natural killer cell, generated from that lymphoid progenitor. What are the majority of the white blood cells? If we were to look at the percentages of what exist, the majority of your white blood cells are neutrophils and then lymphocytes. So you have a lot of neutrophils, and they act fast. And so they are one of the first cells that come and try to fight an infection. And they're important phagocytic cells. They can eat and destroy microbes and pathogens. What are main functions of the white blood cells? So if we look at these different white blood cells, and if we start on the myeloid progenitor side and look at the granulocytes that are there, the neutrophil is a phagocytic cell, so it can eat and destroy microbes. It has multiple things inside of it that can help it do that, and there's a few listed here. Antimicrobial proteins harm pathogens. It can also release things that are important for inflammation and increasing vasodilation, which is helpful in promoting other cells to be recruited to the area, letting the body know that there's inflammation in that area. Eosinophils are important. They have important antiviral activity. They're also important against worm infections, and they are also able to increase vasodilation. Basophils are inflammatory, and they can promote vasodilation and smooth muscle activation, as well as mast cells. Mast cells are an important cell that we'll talk more about when we look at allergy later on in the course. Some of the other cells on the myeloid progenitor side are the monocytes, which are what they look like in the blood, and then they can go into the tissue and become a macrophage, and so they look a little bit different. They are phagocytic, and so they can eat and destroy pathogens as they come in contact with them. They can also present antigens to T cells. So macrophages are antigen presenting cells. So are dendritic cells. They are able to even travel towards a T cell, so they're a professional antigen presenting cell. If we go over to the lymphoid progenitor side of the hematopoietic stem cell chart, we have lymphocytes, which are T cells or B cells. And if we look at the T cells, we can divide them into two main groups. There are other groups that we'll look at later on, but the two big groups that we'll look at are the T helper cell and the cytotoxic T cell. And we can tell it's a T helper cell because it has CD4 on its surface. And we can tell it's a cytotoxic T cell because it has CD8 on its surface. And then B cells have membrane-bound immunoglobulin for its B cell receptor, and it, when it's activated, can then become a plasma cell which secretes antibodies rather than having a B cell receptor on its surface. Down here are the innate lymphoid progenitor cells, and a natural killer cell is an example. And it's an important cell because it can notice if something is wrong with a host cell, and it can often kill host cells that are infected that cytotoxic T cells aren't able to kill. So what are some of the important CD molecules we should know? The CD molecules stand for cluster of differentiation, and we're going to, throughout the course, look at more of them, but here's a few of the ones that we should first start getting to know. All T cells have CD3 on their surface. Some T cells, so the T helper cells, have CD4. The cytotoxic T cells have CD8. And then natural killer cells can be identified by having CD16 on their surface. B cells have CD19, CD20, and CD21. And CD21 is also that complement receptor 2, which we look at later on. CD28, we haven't talked about a lot yet, but we will later on. It is important for as a co-stimulatory molecule on T cells, and it binds to B7 molecules, which are on antigen-presenting cells. Another one that we'll look at more later on, it's important for T cell signaling. So CD40 is on the B cell, and it can interact with CD40 ligand on T cells. And we'll see those interactions more later on in the course. How do B cells recognize antigen compared to T cells? So B cells bind to soluble antigen and get activated through the B cell receptor, which is a membrane-bound immunoglobulin. So the ends of the antibody bind to the antigen, and that activates the B cell. And cross-linking of multiple of them is able to produce a signal, whereas just one antigen binding to the receptor doesn't actually do it. It does require cross-linking. T cells, however, don't recognize antigen that's floating around extracellularly like this. It needs to be presented antigen in the context of MHC. And this example is showing us how the T helper cell recognizes a peptide interacting with MHC class 2. So the T cell receptor recognizes this peptide with MHC class 2. And CD4 also helps identify that this is MHC class 2 that it's recognizing. What are the primary lymphoid organs? So this is where the immune cells are developing. And the B cells develop in the bone marrow. And the T cells originate in the bone marrow, but then they migrate to the thymus to mature. So two main locations where immune cells develop are the bone marrow and the thymus. And the T cells are the ones that mature in the thymus but originate in the bone marrow. What are the secondary lymphoid organs? So we looked at a few, and they're shown here in blue. The secondary lymphoid organs is where you get an immune response initiated. So the tonsils, lymph nodes, spleen, Peyer's patches. These are a few different examples of a secondary lymphoid organ. Over here is a diagram of what a lymph node actually looks like. And you have antigens coming in through these afferent lymphatics and then leaving through the efferent lymphatic vessels. And then there's naive lymphocytes, meaning they haven't seen antigen yet, and they come in through these high endothelial venules into the lymph node. And then in the lymph node, there are B cell zones, which make up a follicle, and then T cell zones in the paracortex. And it's in the follicle where you have B cell activation. And if they're maturing and after being activated, they'll generate this germinal center there. You also get memory cells generated in the lymph node. The spleen is where you get blood-borne pathogens brought in. So this is where you get basically the filtering of things that are in the blood. And this is kind of like a big lymph node, but for the blood. And other tissues that are where you can get an immune response activated are these mucosa-associated lymphoid tissues. There's gut-associated lymphoid tissues, since this is a location near where pathogens are coming in, so near the mucosa, and they have access to some T cells and B cells just near there. They may then, there is able to be presentation with antigen presenting cells to the T cells here, but they may still need to go to a lymph node to locate the right cells to be activated. So primary lymphoid organs, that's where you get the cells developing, and the secondary is where you get an immune response initiated. What is a cytokine? Cytokines are proteins that can signal by binding to a receptor, telling another cell that they should do something different. When they bind, they will be non-covalent binding, but they can bind with high affinity, and it induces a change in the cell that it's binding to the receptor on. So how do the cytokines function? They can act on the same cell that made it, so they can act in an autocrine action, or on a cell nearby, so a paracrine, or on a cell that's far away, so an endocrine fashion. And there are different aspects or properties of cytokines. So they can be pleiotropic. For example, they can have actions on multiple different cells. They can also be redundant in that many of the different cytokines can do the same thing. Or they can act together synergistically to change how a cell is being activated. And they can also antagonize one another. So some of the cytokines will activate one thing to happen, so B cells to be activated to make IgE, whereas another one, such as interferon gamma, inhibits that, so they can antagonize the actions of one another. They can also work through cascades. So one cytokine can act on one cell to do one thing, and then that can cause another cell to do something else. What do the IL-1 cytokines promote? In general, they promote inflammation. They can also act locally to help other white blood cells coming into the area, and so they can have effects on the permeability of the capillaries. They can also go to the liver and promote acute phase protein to be produced, and they can help activate adaptive immune responses. What's unique about the class 1 cytokine receptors and how do they signal? So they tend to have multiple subunits in their receptors, and some of them have common subunits within multiple different cytokines. And that the fact that they often have a common subunit may mean that those cytokines may have some similar functions in how they work. And the class 1 cytokines act through JAK-STATs. So JAKs interact with the receptor on the intracellular side, and when that receptor gets bound to its ligand, it gets phosphorylated. And then when those JAKs gets phosphorylated, they phosphorylate STATs. And when the STATs get phosphorylated, they dimerize, and then they go to the nucleus and become a transcription factor that can change which genes are transcribed. What are the main roles of interferons? So we're going to focus on type 1 and type 2 interferons. And the type 1 interferons are alpha and beta, and they have important antiviral effects. They can tell a nearby cell that there's viruses in the area and to protect themselves from allowing viruses to replicate in them. The type 2 interferons are interferon gamma, and they have important roles in the adaptive immune response. They also signal through JAK-STATs as well. What is TNF? So TNF is tumor necrosis factor. It's important for regulating development and effector function, and it can bind to the TNF receptor, which trimerizes when TNF binds. Signals through NF-κB, which is a transcription factor, and it's pro-inflammatory. TNF alpha is a pro-inflammatory cytokine. Are IL-17 cytokines pro-inflammatory? So we looked at these, and there was a chart, and most of them, not all of them, but most of them are pro-inflammatory in general. What are the chemokines' main role? So they direct white blood cells to move. They direct leukocyte migration. They cause the movement of cells. And they are G protein coupled receptors, and they are basically they're named by where they have these cysteine residues. So they might be CXC or CC based on these conserved cysteine residues. What are the main components of the innate immune response? So if we think about the innate immune response, first trying to block things from getting in, blocking pathogens, and then if the pathogens do get in, trying to destroy them. And if they can't destroy them, they then want to activate. The innate is important for activating the adaptive. So here is showing you an epithelium, which is a barrier. So it's a physical barrier. The epithelium is a physical barrier. And there's also chemical barriers here. So the acidic pH, antimicrobial proteins and peptides that are released, those peptides, antimicrobial peptides, can lyse microbes. They can poke holes in them and kill them. And so these are barriers that are on our skin, in our mucosa, that are trying to act to help prevent infections. If the pathogens do get through those barriers, they can be eaten and destroyed by phagocytic cells. So macrophages are important phagocytic cells. Neutrophils are also phagocytic. If that's not enough, they will present antigen to T cells. So macrophage and dendritic cells and B cells are important antigen presenting cells. They can move from the local area to a lymph node and try to find a T cell to present antigen to. If we look at the cells that are in the innate immune response, those macrophages and the dendritic cells, they have pattern recognition receptors that recognize conserved patterns on pathogens. So those are called pathogen-associated molecular patterns. So on these pathogens, they have these conserved patterns that these recognition receptors can detect them. So some of the pattern recognition receptors are called toll-like receptors. So what do the toll-like receptors recognize? So this is some of the general characteristics of what some of these toll-like receptors can respond to. So a few of them that I want you to know are TLR4, because it can recognize gram-negative bacteria. We also know that TLR2 with TLR1, a heterodimer, can recognize bacteria or parasites. TLR2 as a heterodimer with TLR6 can recognize gram-positive bacteria and fungi. TLR5 recognizes flagellin from bacteria. And then, so those are a bunch of them that are on the outside of a host cell. There are some receptors that are inside the host cell but on an endosome or lysosome. And so many of these are able to detect viruses as well as bacteria. So the TLR4 recognizes viral proteins, and then TLR3, 7, and 8 recognize viral nucleic acids. So viral double-stranded RNA is recognized by TLR3, and then viral single-stranded RNA is recognized by TLR7 and 8. And then bacterial or viral DNA is recognized, or plasmodium is recognized by TLR9. What are some of the differences in TLR signaling in response to bacteria versus viruses? So if we kind of look at the pathways, this is an example of the pathway in response to bacteria that's extracellular. And this pathway over here is responding to viral RNA that's in an endosome. And so one of the main things that is different between these two is that you get interferons generated after a viral response, which makes sense because we know those type 1 interferons are important in telling nearby cells that there's a viral infection. In both pathways, you get the activation of the NF-kappa B transcription factor, and those can have that transcription factor as well as AP1 can have important upregulatory effects on the gene expression of multiple different cytokines and chemokines and other antimicrobial proteins. What pattern recognition receptors recognize fungal cells, and what do they activate? So the pattern recognition receptor that recognizes fungal cells, one of them is the C-type lectin receptors. This is showing you dectin-1, which can recognize fungal cells. And it activates pro-inflammatory cytokines to be generated. What pattern recognition receptor detects bacterial cell walls that are inside of a host cell? So these are the NLRs, or nucleotide oligomerization domain receptors. So this is inside of a host cell. It can detect detect bacterial cell walls. What pattern recognition receptors recognize viral double-stranded RNA inside of a cell? And so these are those RIG1-like receptors, which can detect the viral nucleic acids. How do type 1 interferons actually inhibit viral replication? So there's a few pathways that can activate in type 1 interferons within those pattern recognition receptors, and they can, once a type 1 interferon is made by one cell, it can act on other cells that have an interferon receptor. And that interferon receptor signals through JAKs and STATs activation, and it does a few things that ultimately inhibit viral replication, and that is including inhibiting translation, inhibiting mRNA, so degrading mRNA, inhibiting assembly and transcription of virus material, and as well as inhibiting translation. So a few ways to basically make it so that a virus can't replicate inside of that host cell. What is phagocytosis? So this is basically the eating and destroying of foreign material. So a phagocytic cell can phagocytose, or basically reach out and grab onto something that is external to it and bring it into a phagosome, fuse that phagosome with a lysosome, and then basically break what it's brought into little pieces. And that can be released through exocytosis, which should be broken down little pieces that are no longer functional pathogens. So phagocytosis is what happens when cells try to eat and destroy foreign material. And macrophages are really good phagocytic cells, and so are neutrophils. What can enhance phagocytosis? So there's a few receptors that are on the surface of our phagocytic cells that actually enhance phagocytosis if they are engaged. So the C-type lectin receptors can enhance phagocytosis, as well as complement receptors. CR1 is one example that we'll look at later on. So if CR1 is bound to complement proteins or components, that will activate the phagocytic cell to eat and destroy that pathogen even more. You can also get an enhancement of phagocytosis through through immunoglobulin Fc receptors. So if an antibody is bound to a pathogen, that can be detected by these receptors on the surface of our phagocytic cells. And so complement receptors and Fc receptors, if they're bound to their ligand, that can enhance phagocytosis. And so they act as opsonins, and they're called opsonin receptors. That's opsonization, making something look better for enhancing phagocytosis, ultimately. What are in lysosomes that can kill microbes that are phagocytosed? So in phagosomes, they have reactive oxygen and reactive nitrogen species that can kill and destroy pathogens. What activates inflammation? So if you have tissue damage or bacteria coming into an area, it's going to cause inflammation, and inflammation is the redness, the heat, the swelling, the pain, and possibly loss of function in a given area. And when these bacteria that come in with a tissue damage, they're going to be detected by the cells that are there. So they're going to be detected by the pattern recognition receptors that are on those cells. And those pattern recognition receptors, when they recognize these pathogens, they're going to signal for that cell to make cytokines. And so cytokines and chemokines will be made and released, triggering inflammation in that area. And that makes it so that the area is, the vasculature is leaky. And it also recruits other cells to come in by chemokine production. And when these cells are going through here, they're going quite fast. But if there's inflammation here, they slow down. And so they adhere to the endothelium that's in the blood vessel. And then they'll extravasate and come out into here. And so the ones that tend to come in first are what's drawn here, these neutrophils, but other phagocytic cells will also come in and try to eat and destroy what's been brought in by this tissue damage to try to heal the area. You can also get, from the cytokines that are released in this area, you can get the activation of the liver to make acute phase proteins. That will also help eliminate the pathogens that have come in here. What do natural killer cells do? They are able to kill altered self cells. So they can sometimes kill a cancer cell or a virus-infected cell, and they detect a change in the surface receptors on a host cell. So if a host cell, for example, decreases its MHC class 1 expression, the natural killer cells can detect that, and they will interact with it by releasing perforin and granzyme and causing apoptosis, or programmed cell death of that cell. How does the innate immune response shape the adaptive response? So when innate immune cells come in contact with pathogen, they have pattern recognition receptors that can detect the type of pathogen. So for example, if this innate immune cell, this dendritic cell, detects that there's bacteria there, it's going to make a cytokine such as IL-12, which is going to promote the type of T cell to become a T helper 1 cell, which is going to make interferon gamma. So it's going to shape the T cell response depending on which pathogens this dendritic cell has come in contact with. For example, if you have a fungal infection, you have those C-type lectin receptors such as dectin-1 that detects that, and it'll make cytokines such as IL-6 and IL-23, which will shape the naive T cell to become a T helper 17 cell, which secretes IL-17, which is pro-inflammatory. If you have a Helminth infection, the type of cytokines that will be made may be like IL-10, and IL-10 is more of an anti-inflammatory cytokine, and it tends to make or generate T helper 2 cells, which are important for B cell responses actually, and they also play a role in allergy. If the dendritic cell came in contact with fungal infection, they may make IL-10 or TGF-beta, which are anti-inflammatory, and they can direct the T cells that are presented these antigens to become T regulatory cells. And T regulatory cells are important because they decrease the inflammation by producing cytokines such as IL-10 and TGF-beta that are anti-inflammatory. So overall, the innate immune response will ultimately shape the adaptive immune response, because what this dendritic cell came in contact with will influence how the T cells are activated. What are the outcomes of complement? So when complement gets activated, the complement proteins are in the blood in an inactivated form. And when they get activated, they can do a few things. They can act, some of the proteins, the complement proteins, such as C3b, can act as an opsonin and enhance phagocytosis. They can act as anaphylatoxins, which cause inflammation and can promote the chemotaxis of other cells to the area. Complement proteins can bind to the bacterial cell membrane and poke a hole in it, so that make a membrane attack complex, so causing lysis of that cell. And there are complement receptors that can bind to some of the complement proteins, which can also have effects such as enhancing phagocytosis, and also have important roles in bringing immune complexes from the blood into the liver to be cleared. So those are some of the main goals of complement. What are the 3 different ways that activate complement, or that complement are activated? The classical, the lectin, and the alternative. And so the classical is one that's connected to the adaptive immune response, because there are antibodies that are bound to a pathogen, and that's what initiates C1q to interact with the antibodies that are bound to pathogen. C1q with R2 and S2 are able to cleave C2, C4, and C2, and this will cause C4b to bind to the pathogen with C2a, making a C3 convertase. So this enzyme here is able to take C3 and break it to C3a and C3b. And C3a will go and be inflammatory, and then C3b can bind to other pathogens over here, or can bind to C3 convertase, becoming C5 convertase. So that's how the classical pathway can be activated. The lectin pathway binds, this mannose binding lectin binds to carbohydrates such as mannose on the surface of pathogens, and a molecule that looks similar to C1q, so the mannose binding lectin is what binds to those carbohydrates. And then there's these mannose binding lectin associated serine proteases, which act similar to what the C1R2S2 did, and they are able to cleave C4 and C2 and generate a C3 convertase, similar to what happened in the classical pathway down here. So the classical pathway is recognizing antibody bound to a pathogen, and then in the lectin pathway, it's a mannose binding lectin that binds to carbohydrates on the surface of a pathogen. And then the third pathway is more spontaneous hydrolysis, and it's due to the fact that C3 can hydrolyze and can be split into C3a and C3b. And then C3b, which is also generated down here from the C3 convertase, it can, with factor B, become a C3 convertase. So in the alternative pathways, the C3 convertase is different than the C3 convertase that's in the lectin or classical pathway. And how this C3 convertase is generated is because the C3b with a cleaved factor B, which is big B, little b, binds together, making it. And it's factor D that cleaves the factor B. And properdin is something that stabilizes this and can help initiate it in some cases. Overall, then you get, in either case, whether it be from this pathway or this one, if you get C3b binding to the C3 convertase, you get what is a C5 convertase. And C5 convertase takes C5 and cuts it into C5a and C5b. And C5a is important for inflammation, so it's an anaphylatoxin, which can cause other cells to come and be recruited to the area, or it can also cause degranulation. And then if it's C5b, if it binds to the pathogen surface, it'll promote ultimately lysis of that cell. So what cleaves C4 and C2 in the classical complement pathway? It is the C1s that cleaves C4 and C2. What resembles C1q in the lectin pathway? So that's the mannose binding lectin, which resembles the C1q. How is the alternative pathway activated? So there's a few ways that we looked at. There can be a fluid phase or a membrane-bound phase, and both of them have this spontaneous change that occurs in C3. And so C3 with this spontaneous hydrolysis of it can combine with factor B. Factor B can be cleaved by factor D, giving you C3 with big B, little b, which becomes a C3 convertase. And so similar can happen on the surface of a cell. And you can also get this stabilized by properdin, or it can be called factor P. And once you get that, you can get a stable C3 convertase, an enzyme that will function a lot longer. You can also get properdin interacting directly with the surface and kind of initiating this to happen. So properdin can be the starting factor, and then you can get C3b with factor B, and then factor D cleaving that factor B, releasing big B, little a, and then you can get C3b with properdin and big B, little b here. How does complement lyse cells? And so that's when the membrane attack complex is made. And so when C5b binds to the surface of that cell, it'll then recruit C6, 7, 8, and then multiple of the 9s, and that's what pokes the hole in that cell. And that hole will cause that cell to lyse. What protects host cells from lysis due to complement? So our host cells have protectin, or CD59, that protects them from C9 poking a hole in our host cells. What else protects host cells from complement? We also have C1 inhibitor, which will cause that serine proteases to dissociate from C1q. We also have decay accelerating factors, which will break down the C3 convertases. So in the classical pathway, we have the decay accelerating factor with CR1, which is a complement receptor 1, and C4 binding protein, which will break down the C3 convertase into C4b and C2a. And then in the alternative pathway, we have decay accelerating factor with CR1 and factor H, which is able to break down that C3 convertase into C3b and big B, little b. We also have factor I that further breaks down C3b and C4b with other molecules. So with MCP or factor H, can break down C3b into smaller fragments, and C4b can be broken down by factor I with the help of MCP, CR1, and C4BP into C4C and C4D. So ultimately, factor I can break down these complement proteins into smaller pieces. What regulates C3A and C5A? So these are the anaphylatoxins, and the C5A is more potent. And they can be regulated by these carboxypeptidases, which inactivate these anaphylatoxins. So they break them down, so they're no longer functional. What do the C3A and C5A? I said they're anaphylatoxins. They cause inflammation. So they can bind to receptors on cells, and such as macrophages and these granulocytes, and they can cause chemotaxis or degranulation as a few main examples of what they can do when they bind their receptor. And so these can be regulated by those carboxypeptidases here. What does CR1 do? So it can help remove immune complexes from the blood. CR1 is on the surface of red blood cells, and it can help bring immune complexes to the liver or spleen so that they can be cleared from the blood. It's also on phagocytic cells, and if CR1 is bound to C3b, it can enhance phagocytosis of pathogen, so improve the eating and destroying of it by phagocytic cells. And the other one that I want you to know that can do that and act as an opsonin as well as the FC receptor. So an FC receptor binds to an antibody bound to an antigen, and that can enhance phagocytosis as well. What does CR2 do? So that's a complement receptor. It's also called CD21, and it's on the surface of the B cell, and it can bind to complement protein C3D. And when C3D is bound to an antigen, and that antigen is bound to the B cell receptor, this can help give a signal to that B cell to help activate it a bit better. Can microbes evade the complement pathway? They can. There are multiple different ways that they can evade. One example is that strep pyogenes have M proteins that can bind to C4 binding protein and factor H, and can actually make it so that C3 convertases decay quicker, even though they're bound to a bacterial surface. How are the B cell receptor genes organized? So the B cell receptor genes have multiple segments encoded in the germline. And there's an example here of the light chain, and an example here of the heavy chain. So the light chain has variable regions and joining regions and constant regions. So different segments that are combined together to give you the light chain of the immunoglobulin that makes up the B cell receptor. In the heavy chain, there are variable regions, which, so there's variable segments, diversity segments, joining segments, and then constant gene segments. And these recombine to give you different heavy chains, which will ultimately give you different B cell receptors. And for each immunoglobulin, there are 2 heavy chains that are identical and 2 light chains that are identical. The light chains could be kappa or lambda though, and they're more often kappa, but they can be lambda if the kappa ones for a particular B cell are not functional. How many different combinations of B cell receptors are possible? So because we have multiple different segments, and in the heavy chain we have V, D, and J, And in the light chains, we have V and J segments, and then we could have kappa or lambda. There are, if you take into consideration all these different combinations of heavy chains and the different combinations that can exist for light chains, and the fact that you can have heavy chains with different light chains, you get over 2 million possible ways to organize these genes, ways that they can be recombined. What parts of the DNA are recognized for recombination? So there are these recombination signal sequences that are on sides of the segments of the genes. So on the top here is the light chain, and on one side of the V segments there are these RSS elements, and on the J there are RSS elements as well, and they recombined in this 12-23 rule. So a 12 RSS element has to recombine with a 23 base pair 1. And in the heavy chain, there are RSS elements on one side of the V segments, on both sides of the D, because the V and the D and the J and then the C have to connect together. So on the V and it's on the D and the J that happen first, and then it's the V and the D. And so this 12-23 rule applies. What enzyme is essential for recombination that is unique to lymphocytes? So all cells of the body have this enzyme, but only lymphocytes express it. And it's this recombination activating gene. There's REG1 and REG2. And this enzyme is important for interacting with those RSS parts of the gene and recognizing and cutting it at the RSS spot. What are the main steps in recombination? So the REG proteins are able to locate a particular V and a J in this particular example and bring them together and cut one side of it, ultimately giving you this hairpin on one side, and then this one will go and reconnect with the other part that's cut off. Then you get at this hairpin, you get this endonuclease Artemis, which can cut in one of 3 different ways. So you can get even more diversity because of the different possibilities of where this enzyme is going to cut this hairpin. And then you have the addition of nucleotides, so P nucleotide addition, if you open this and it has an overhang. So you'll get a few nucleotides added in here depending on how it cuts, how Artemis cuts it. And then we have enzymes that are important for sealing this DNA back together. So DNA ligase 4 and XRCC4 can glue this basically back together. So what contributes to the diversity in the B cell receptor? So those multiple gene segments that we looked at, and they can be combined, that gives us diversity of about more than 2 million different possibilities. When you combine the different gene segments and the fact that you have different heavy chains and light chains that are going to combine, then we get even more possible diversity generated in these naive B cells because we get nucleotides that are added in at that hairpin structure, which depends on where Artemis will cut it. We also have exonuclease trimming, so sometimes we lose nucleotides at junctions. And then there's also TDT activity, which adds in non-templated nucleotides. And this doesn't tend to occur in the light chain, but does happen more in the heavy chain. So what is allelic exclusion? This is the fact that, or this describes the fact that you only get one allele expressed for the heavy chain and one allele expressed for the light chain for a B cell, even though there are multiple that could be used. And so when a B cell is a progenitor B cell, the first thing that happens when it's recombining its genes is that you get the heavy chain recombining first, and it goes from the D segment recombining with the J segment. And if that recombination is proceeds, it'll then go from the V to D, and then you'll get a heavy chain that's brought to the surface of that cell with a surrogate light chain, if it's a productive allele. If it's productive, it means that it makes the heavy chain and it goes to the surface and it has all the parts of it that are needed. If it's non-productive, meaning say there's a stop codon in the middle of it and the gene is, when it's transcribed and then ultimately translated, it doesn't make functional protein, it would be non-productive and it has another try. So it could try the second allele, because there's two alleles for the heavy chain, and that may be productive. If it's not, though, that cell will then be destined to die because it's not going to put a cell out of the bone marrow. It's not going to take a B cell unless it has a B cell receptor on its surface. So if it gets a productive B cell, gets a productive heavy chain, it's called a pre-B cell receptor because it's matched with a surrogate light chain. And then it will rearrange the light chain genes, and it usually does the kappa genes first. So it's going to try the kappa genes, the first allele, with a V and J rearrangement, and if that's productive, it will put that B cell receptor on the surface of it. If it's not productive, it will try the second kappa allele, and if that's productive, that new recombined immunoglobulin will go to the surface. If it isn't productive, however, it has two more tries because it can try the lambda genes, and there's two alleles for the lambda light chain gene. And if that one is productive, it will put a B cell receptor on the surface based on that lambda gene that was recombined. If it's not productive, though, it has one more try because it has a second allele for the lambda chain to try. If that second lambda gene isn't productive, that cell will then be destined to die because it doesn't want to release any B cells that don't have a B cell receptor on its surface. And so this process ensures that only one allele is expressed for the heavy chain and only one allele is expressed for the light chain. If a B cell receptor binds to self-antigen, how can the B cell receptor be salvaged? So say you got your productive B cell receptor, whether it's this one, or this one, whichever one was productive. Say it actually detects self-antigen. That's not something that we would want. So how can that B cell receptor be salvaged? You can actually have receptor editing. So the segments that are left in there that didn't get removed by the recombination initially, you can then try some of the other segments that are there, and it can maybe salvage that B cell receptor by editing the receptor that's there and try again. What is the mechanism of making B cell receptors that are IgM and or IgD? So IgM or IgD are the B cell receptor. It's the type of immunoglobulin that will go on the surface based on the constant region of that B cell receptor. And it decides if it's going to be a IgM or IgD based on splicing of the mRNA. So the recombination events we were just talking about are at the DNA level. This splicing of the mRNA is at the RNA level. And so you can get IgM mRNAs spliced together, or you can get IgD mRNAs generated from the same gene. What controls whether antibodies are membrane bound or secreted? This is also through splicing at the mRNA level. So there are secretory segments or membrane bound segments that will tell basically the mRNA to make a protein that is going to be secreted or going to be membrane bound. And so the membrane bound form is what happens in a B cell that has not yet been activated, and this is spliced this way if the B cell has been activated. What does the T cell receptor look like, and how do the genes look? So the B cell receptor looks like this. It's most often alpha and beta chains that make up the B cell receptor, and it does cross the membrane, but has a very short intracellular domain. There can also be gamma delta T cells, but the alpha and beta are more common. And the T cell receptor, the alpha chain, has V and J segments. Similarly, the gamma has V and J segments, whereas the beta and delta have V, D, and J segments. Do the T cell receptor genes go through recombination? Yes, they do, in a very similar manner to the immunoglobulin genes. They do go through recombination, so they use those same enzymes, the REG1 and 2 and the Artemis and the ligase. That has a very similar process for generating the T cell receptor. Which T cell receptor chains look like the heavy chain? So the heavy chain has V, D, and J segments, and in the T cells, the beta and delta regions, the chains, have V, D, and J. So the T cell beta and delta chains look most like the heavy chain. When the T cell is activated in the T cell receptor, when the T cell is activated, is the T cell receptor secreted like the inactivated B cells? No, the T cell receptor does not have a secreted form. The B cells, when they're activated, they start secreting antibody rather than having membrane-bound antibody. After activation, does the T cell receptor go through somatic hypermutation like inactivated B cells? That is also no. The T cells do not go through somatic hypermutation, but the B cells do, and this is a way for them to have an antibody that binds a bit better. How do T cell receptors recognize antigen? So they need to see antigen in the context of MHC. So T cell receptors, those alpha and beta chains or the gamma delta, they only recognize antigen presented in an MHC molecule. And this here is showing us the MHC molecules, MHC class 1 and MHC class 2. And MHC class 1 has 3 extracellular domains and crosses the membrane once, and is associated with beta-2 microglobulin. Where the antigen binds is in between alpha 1 and alpha 2 of the MHC class 1 molecule. And it's a short peptide. It's about 8 to 10 amino acids long that can bind to an MHC1 molecule. MHC class 2 crosses the membrane twice, and it has 2 extracellular domains in the alpha chain and 2 extracellular domains in the beta chain. And in the peptide binding groove, it's made up of alpha 1 and alpha 2, alpha 1 and beta 1. And the amino acid sequence is a bit longer. I think it's between 13 and 18 amino acids long. So it's a bit, the size of the peptide is a bit longer that can bind to the MHC class 2 molecule. Why are polymorphisms beneficial in MHC genes, and how many different MHC1 and 2 are expressed in most people? So, having polymorphisms are beneficial in MHC genes because this means that there are many different allelic variants in humans, and so in general it increases the chances that there will be people that can respond to a pathogen if we have differences in where or how our MHC bind to peptide. And how many different MHC1 and 2 are expressed in most people? Most people have 6 different class 1 molecules and about 12 different class 2 molecules, and that's because you get 3 class 1 from each parent, so you get 6 class 1 total, and you get about 6 different class 2 molecules, and so you get 12 total because you have 1 set from each parent. And the polymorphisms that exist, they're shown here in these black bars. They tend to be where the peptide binds in the peptide binding groove, and that is beneficial because that means that there are different, or there's changes or differences in the way that the peptide can bind to the MHC molecules. What are the MHC1 and 2 molecules called in humans? So they're the HLA molecules, and in MHC class 1, there's HLA A, B, and C. And for MHC class 2 molecules in humans, there's HLA DP, DQ, and DR, and there's an alpha and beta chain of each. So that gives us the 6 different possible from one parent right there. This would be one gene. How are MHC genes inherited and expressed? So they are inherited in groups called haplotypes, and they're expressed codominantly. So you'll have the expression of both your maternal and paternal MHC molecules. How do MHC genes affect transplants? So it's really important, and this is what physicians who are looking at MHC molecules examine the MHC molecules. And so transplants only work if someone have very similar MHC genes. And so this experiment on the left here is looking at mice, and it shows you how if the donor graft doesn't have the same genetics or in the MHC genes as the recipient, it's going to be rejected. So this individual can donate to a genetically similar individual, but not to an individual that has the MHC gene that's different. And it's showing you that for 2 different strains of mice. And then it also shows you that if you make a progeny of that mix of that 2 mice, that progeny isn't able to donate to the mother or the father because they are combined, and it will be rejected because it'll see the other strain or the other MHC molecules that are there. What does polygenic mean? It means that there are many genes that do the same thing, but they have a fairly different structure. And so this is showing you in a mouse model, but you see that there are multiple different, the idea of polygenic is shown here because you have multiple different MHC class 1 molecules here, and they do the same thing, even though they're structurally a little bit different. And down here you see that for the MHC2 molecules. So there, it also highlights that they're co-dominantly expressed. So you get one haplotype of your MHC molecules from your mother and one haplotype from your father, and both of them are going to be expressed on the surface of your cells. What antigens are presented on MHC 1 and 2? So you get the expression of intracellular antigens predominantly on class 1 MHC molecules, and you get the expression of extracellular antigens on MHC class 2. And so this diagram kind of highlights that idea that things that are inside of a cell will be broken down and processed and presented in MHC class 1. So you'll get viral antigens expressed on MHC class 1. You'll get intracellular bacteria expressed on MHC class 1. Extracellular antigens, so extracellular bacteria, or a virus that has not infected the cell yet, may be brought in to an endosome, and this can then be presented, pieces of it, in MHC class 2 molecules. So something that antigen presenting cell came in contact with, something it phagocytosed and brought into it, will be presented on MHC class 2. And MHC class 2 will be presented to T helper cells that are CD4 positive, whereas MHC class 1 presents antigen to those cytotoxic T cells. So what cells have MHC1 and which ones have MHC2 and which ones have both? So MHC class 1 is on all nucleated cells. So red blood cells don't have MHC class 1, but all nucleated cells basically have MHC class 1. That's important so that it can basically constantly be showing some of its proteins and be saying, am I a healthy cell? Are these proteins that are inside of me healthy? So all cells of the body that are nucleated can do that. Then MHC class 2, which is the one that presents exogenous antigen, it's only on antigen presenting cells. And so you have macrophages, B cells, and dendritic cells that are antigen presenting cells. There are some non-professional cells that can present them, and that's important for the generation, basically for kind of training and generating tolerance. But for our purposes at this particular time, we're going to focus on the fact that MHC class 2 is predominantly on antigen presenting cells. Can the number of MHC molecules change on a cell? They can. During infection, you may have an increase in MHC molecules on the surface of cells. You can also have viruses that interfere and they can maybe shut down MHC class 1 expression. So that's their way of evading the adaptive immune response. They can evade cytotoxic T cells by shutting down their expression of MHC class 1. You can also get cytokines such as interferon alpha and TNF alpha to increase MHC expression, whereas other molecules that are generated sometimes, such as corticosteroids or prostaglandins, will downregulate MHC expression. So their expression can change depending on what's going on around the cell. Do T cells recognize MHC from a genetically different individual? They don't. They are self-restricted MHC alleles. So a T cell from one strain of mouse will only recognize antigen in context of antigen-presenting cells that are the same genetic strain. It won't detect the MHC molecules of a different strain mouse. Now it will if it's a mouse that is a progeny crossed with it. And so this is one way that this is one experiment looking at the T cells from different mice and the antigen presenting cells from different strains of mice, and identifying when you get T cell proliferation occurring. And they realize that it's only when you have the T cells recognizing self MHC molecules, like from the same animal, or genetically the same animal. Can T cells detect whole antigen? They can't. So if you have antigen presenting cells and you fix them so they're basically dead, but they're still there, not broken apart, and you put antigen in, you're not going to get T cell activation because the T cells need to recognize small pieces of the peptide in MHC molecules. If, however, you allow that antigen presenting cell to process the antigen and put it in its MHC class 1 and then fix that antigen presenting cell, you will get proliferation of the T cells. If you take fixed antigen presenting cells and give them broken down antigen, so you take that antigen and you break it into little pieces, it can swap out what it has there and put in the new antigens to activate that T helper cell. So T cells only recognize antigen peptide fragments presented in MHC molecules. They don't recognize whole antigen. How does antigen get processed and put onto MHC molecules? So antigen gets processed from inside the cell or outside the cell. So if it's an endogenous antigen, so a protein, some of them will get ubiquitinated and destined to go to the proteasome to be broken down into smaller peptide fragments. Those peptide fragments can be brought into the rough endoplasmic reticulum with TAP. And then once it gets in there, it will be these peptides will be broken down even smaller with the molecules that are in here. There are chaperones that help MHC class 1 before it gets its peptide bound to it. So MHC class 1, which is right here, with the help of calnexin, calreticulin, and taphazin, it helps stabilize it with the ERP57. And then with the beta-2 microglobulin, these will dissociate when the peptide is broken down and put into the top here. So kind of the chaperones help keep this stable until the peptide is able to be broken down into a smaller piece and put in here. This will then go from the rough endoplasmic reticulum through the Golgi and then to the surface of the cell, presenting this antigen which came from inside the cell from an endogenous protein. Exogenous proteins, so things that come in from outside of the cell, exogenous antigen, will be presented in MHC class 2. When MHC class 2 is made in the rough endoplasmic reticulum, it's initially stabilized by this invariant chain, and that invariant chain will help bring it to a phagosome. It'll get cleaved to clip because it will get cleaved once it's as an endosome. And when a phagosome combines with that endosome, it will swap out some of the peptide pieces that were broken down from an exogenous antigen. They'll swip out the clip and put in the peptide. and then this will go to the surface of the cell. So you'll have presentation of antigen from an exogenous source in MHC class II. Importantly, you do get cross-presentation in dendritic cells of exogenous antigens into MHC class I and onto the surface of the cell. And this is so that you can get activation of CD8 positive T cells. So even though this antigen presenting cell may not be infected with a particular virus, it may bring in exogenous antigen or viruses from that it's found and be able to present it to CD8 positive T cells to activate them to then go out and find that type of pathogen that may infect cells. So why do dendritic cells cross-present antigen? It's so that they can prime those CD8 positive T cell responses. So this is what normally happens. A T helper cell will be presented antigen in MHC class II, and that antigen will have come from exogenous source, so brought inside that cell, and clip will flip out, and then this exogenous antigen will be combined with the MHC class II and presented on this T cell to activate that T cell. Some exogenous antigens in dendritic cells will be cross-presented to be put inside of MHC class I molecules so that you can get the activation of naive cytotoxic T cells, so that you can have this naive T cell be able to be activated to then go out and find infected cells. And so if this didn't happen, you wouldn't, unless this dendritic cell was infected, you wouldn't necessarily have a way of activating this cytotoxic T cell. So this is an important way. This cross-presentation seems to only occur in dendritic cells, of taking something that's exogenous and putting it in an MHC class I. So majority of the time, the exogenous antigens are presented in MHC class II, and endogenous antigens are in MHC class I. However, dendritic cells are able to cross-present exogenous antigens into MHC class I, and that's so that they can activate these naive CD8 positive cytotoxic T cells. How are lipid antigens presented to T cells? They are presented in not by normal MHC molecules, but the CD1 molecule, which is the MHC class I related protein. And so lipids, which are non-peptide antigens, wouldn't be able to be presented in a normal MHC class I or II, but they can be presented in the CD1. So I hope this helps. Focus where you need to study.